Okay, let's unpack this. Think about being in the middle of something complex. Maybe you're reaching for something, walking on tricky ground, even just typing fast.
Mhm.
What happens if there's a tiny bump, or you slip for a second, or hit the wrong key? Usually you don't just stop.
Right, you correct almost instantly.
You correct, yeah, almost without thinking. And the overall plan, whatever you are doing, it stays on track.
It's that resilience, that ability to handle minor disruptions. It's really quite remarkable when you stop and consider it.
Exactly. How does the brain actually pull that off? How does it keep a plan stable even when things go a bit wrong for a moment?
Well, that inherent stability is, one of the brain's most amazing features.
Yeah.
And frankly, a pretty fundamental puzzle in neuroscience. How does it generate and hold on to a stable plan, filter out the noise, and bounce back from being knocked off course?
And that is exactly what we're diving into today. We're focusing right in on the brain's motor planning system to try and uncover the mechanisms behind this robustness.
Yeah.
And our sources today are really centered on findings from one specific brain region in mice, the anterior lateral motor cortex, AL.
That's right, ALM. We're taking a close look at some really detailed scientific work here, mostly from cutting-edge research using mice. And the scientists were clever. They combined behavioral tasks with, neural recordings and these powerful techniques like optogenetics.
Ah, the light swishing thing.
Exactly, using light to temporarily mess with brain activity,
Yeah.
to directly poke at this planning system and see how it handles disruption.
Okay, so our mission for you, listening in, is to unpack what these studies tell us about how the brain makes sure its motor plans are so resilient, how they bounce back, and, what does this reveal about how this key planning area is actually built?
Right, so let's set the scene first. What was the actual task the mice were doing, and what's the ALM's job in all this?
Okay, the task, it's pretty standard for this kind of work. A mouse uses its whiskers, which are incredibly sensitive, right? But to figure out where a little pole is, left or right. Then the pole disappears. And there's this delay, maybe a second or so, where the mouse just has to remember the location and get ready.
Hold that thought.
Hold that thought, exactly. Then a sound cue tells it, go, and it reports the location by licking either a left spout or a right spout for a reward.
Got it. And the ALM, this brain region, we know
Yeah.
it's critical during that delay period.
Yeah, that's its time to shine.
It's actively planning the upcoming lick. Which direction? It's holding that get-ready signal.
So when the researchers actually look at the neurons firing in ALM during this planning time, what do they see? What do the patterns look like?
Oh, they see some fascinating stuff. Some neurons show what's called persistent activity. They just stay active, more or less steadily, through the whole delay, kind of like holding on to a piece of information.
Okay.
Others show ramping activity. Their firing rate gradually climbs up, or sometimes down, almost like they're building up anticipation or momentum towards the actual movement.
Building up.
Right. And the crucial thing is, these patterns are selective. They look different depending on whether the mouse is planning lick left versus lick right. That selective activity is the brain's plan.
Okay, so you've got this selective plan brewing in the ALM. How did they then, deliberately mess with it to test how robust it was?
They use that tool we mentioned, transient photoinhibition. It's a really neat technique. You use light delivered precisely to temporarily and reversibly silence neurons that you've engineered to be light sensitive.
So like a remote control off switch for brain cells?
Pretty much, yeah. A temporary one. And in one of the key experiments, they aimed this off switch at the ALM. They transiently silenced a big chunk, almost 60% of the ALM neurons, but only in one hemisphere.
Just one side.
Just one side, yeah, for less than a second, right in the middle of that planning delay.
Wow, okay. So for that short time, one side of this really important planning area was basically offline.
That's the idea. They confirmed the activity in that targeted spot dropped dramatically when the light was on.
Right. But here's the kicker. This is where the resilience part comes in. What happened to the activity after they turned the light off? Did it just stay off?
No. And this was amazing. The activity recovered, and it recovered fast.
Yeah.
And for those neurons that were ramping up, the ones building momentum,
Yeah.
they didn't just start ramping again from where they were stopped. Their firing rate actually accelerated after the silencing ended. It was almost like the network realized it was behind schedule and sped up to catch up to where it should have been.
Whoa, like hitting the gas pedal after a stall. That's incredible. But was it just like general activity coming back online? Or did the plan itself, left versus right, also come back?
That's the absolutely critical point. It wasn't just noise returning. The selectivity, the difference in activity that signals left plan versus right plan, that also snapped back remarkably quickly. We're talking under a second after the light went off, most of that directional signal was back.
Okay, so the brain didn't just switch the lights back on, it actively restored the meaningful pattern. That's huge. And what about the mouse's actual behavior? Did this temporary zap to one side mess up its licking?
Well, this tells you a lot about the system's buffers. If the silencing ended early enough in that delay period, giving the brain a bit of time, maybe half a second or more, before the go signal, then the mice's behavior was mostly fine. They still licked the correct spout. The system compensated.
Amazing.
However, if they ended the silencing right before the go cue, leaving basically no recovery time, then yeah, it did cause a problem. It biased the mouse towards licking the side that wasn't silenced.
Ah, okay. So timing matters for recovery.
Exactly.
Mm.
And they also mentioned that briefly boosting activity on one side showed a similar kind of recovery dynamic. The system seems built to stabilize against various kinds of brief disruptions.
Okay, so hitting one side temporarily, the system can handle it neurally and behaviorally if it gets a moment to recover. That alone says something powerful. But, okay, the obvious next question. What if you hit both sides at once?
That was the crucial comparison, yes. They ran experiments where they silenced ALM in both hemispheres simultaneously, again for just a short period.
And the behavior, was it just twice as bad, or?
It was way more than twice as bad. The contrast was really stark. Bilateral silencing hitting both sides led to a severe breakdown in the task. The mice's accuracy just plummeted, often down near chance levels, like they were just guessing.
Wow, so much worse than just hitting one side.
Significantly worse. Even if the total area silenced bilaterally was comparable to, or even a bit more than, the unilateral silencing area, it showed it wasn't just about the amount of tissue offline, but something about having a functioning counterpart.
And neurally, what happened inside the ALM when both sides were hit?
This is really the key to understanding why bilateral was so much worse. After the bilateral silencing ended, the overall average firing rate of the neurons did recover.
Yeah.
Just like in the unilateral case, the neurons started firing again.
Okay.
But, and this is the crucial difference, the movement selectivity, that vital pattern distinguishing left plan from right plan activity, it failed to recover after the bilateral silencing.
So the activity came back, but the information was gone, scrambled.
Precisely. The signal telling the mouse what it planned to do was lost, which explains why their behavior fell apart.
That failure really hammers home that it's the pattern, not just the volume of activity that counts. You mentioned they use this cool way to visualize these patterns in activity space.
Exactly. It's a really useful way to think about it when you're recording from lots of neurons at once. Imagine each neuron's activity is a dimension on a graph. With hundreds of neurons, you get this incredibly high-dimensional space.
Hard to picture, but okay.
Yeah, but the state of the whole network at any single moment is just one point in that space. And as the mouse is planning, that point moves, tracing out a path, a trajectory. Different plans, like lick left versus lick right, follow different trajectories.
And they could find the specific direction in that space that mattered most.
Yes, they identified what they called the coding direction, or CD. Think of it as the specific axis within that huge space where the activity pattern's most clearly separate based on whether the plan is left or right. It's the dimension carrying the most behaviorally relevant signal.
Gotcha. So back to the silencing experiments. When they silenced just one side, what did that look like in this activity space?
Okay, so the silencing pulse would temporarily knock the activity point off its normal path. It moved away from where it should be. But, and this is the cool part, after the light went off, the activity point quickly moved back towards, and then continued along, the correct trajectory. Especially, it recovered its position along that crucial coding direction.
It found its way back onto the right track.
Exactly. It corrected itself.
And bilateral silencing, how did that look?
Fundamentally different. The bilateral hit persistently messed up the trajectory. And critically, it specifically corrupted the signal along that coding direction. Those distinct paths for left and right that normally separate along the CD, they just got blurred or completely lost. The distinction vanished.
Yeah. And the sources also mentioned other activity patterns, right? Things that weren't this coding direction.
Yeah, that's interesting too. They found other sort of dimensions or modes of activity. There was one they called a persistent mode. This one was affected by unilateral silencing, but it didn't seem to predict the behavior very well.
Less important, maybe?
Possibly. And even more intriguingly, there was a ramping mode, a general ramping up of activity, not selective for direction, that seemed robust to all the perturbations. Even the bilateral ones couldn't stop this general ramp.
Huh. What might that mean?
Well, it suggests that maybe part of that ramping signal isn't generated purely within the local ALM circuits that create the selectivity. It might be a more general get ready or timing signal coming into ALM from somewhere else, something less fragile.
Okay, that makes sense. So the really key takeaway here for you listening is that when only one side got hit, the brain's recovery wasn't just like hitting reset. It specifically seemed to prioritize fixing the signal along that coding direction, the pattern that actually mattered for the plan. It was a smart, targeted recovery.
Which brings us right to the main puzzle, doesn't it? If unilateral hits get fixed, but bilateral ones don't, and the crucial selective signal is what fails after bilateral silencing, what's actually doing the fixing in the unilateral case? Where's the correction signal coming from?
Well, given the setup, it almost has to be the other side, right? The hemisphere that wasn't silenced.
That was the strong hypothesis, and the researchers designed a really elegant, if drastic, experiment to test it directly. They surgically cut the corpus callosum.
Wow, the main highway between the two hemispheres.
Exactly. Specifically targeting the connections relevant to ALM.
Okay, that sounds pretty major. Did just cutting that connection mess up the mice's normal behavior or activity before they did any silencing?
Surprisingly, no, not really. Their baseline performance on the task and the normal planning activity in ALM, it was largely unaffected just by cutting the connection.
Interesting. So the hemispheres can kind of do their own thing, planning-wise, under normal conditions.
It seems so, yes. They don't appear to need constant moment-to-moment correction from the other side when everything's running smoothly.
Okay, but then they did the unilateral silencing again after cutting that connection.
Yes, and this was the absolute clincher. When they performed the same brief, one-sided photo inhibition after the corpus callosum was cut, the results were completely different from before the cut.
How so?
The outcome looked exactly like the bilateral silencing results in intact mice. Behaviorally, performance plummeted severely. And neurally, the critical selectivity of the left versus right signal failed to recover in the silenced hemisphere.
So cutting the communication line between the hemispheres made a single-sided hit just as bad as hitting both sides simultaneously.
Precisely. It provides really compelling evidence that the input coming from the unperturbed opposite hemisphere, traveling via the corpus callosum, is absolutely necessary for the silenced hemisphere to get back on track and recover its specific, behaviorally crucial planning signal. The healthy side rescues the injured side.
That is a seriously profound insight into how the brain builds in redundancy and uses teamwork. Did they try to build computer models to see what kind of circuit could actually behave like this?
Yes, that was another important part of the research. They turned to computational modeling to try and figure out the underlying circuit principles that could explain this kind of robustness.
And I guess simple, standard models didn't quite capture it.
Correct. They tried various standard types of network models, things like basic attractor networks or integrator circuits, or just generally trained recurrent networks that are designed to hold on to activity patterns. These simpler sort of monolithic models, while they could produce stable activity, they generally failed to replicate this specific recovery dynamic. When they simulated silencing a subset of their artificial neurons temporarily, the model's activity didn't bounce back correctly, especially not the selective part. They just didn't show that resilience.
But some models did work. What was different about them?
Yes. The models that did successfully capture the key experimental findings had a specific kind of structure, a modular architecture.
Modular, like building blocks.
Kind of, yeah. They structured the model as two distinct but interconnected modules, explicitly representing the two brain hemispheres. And crucially, they included specific connections between these modules, designed in a way that allowed one module to provide corrective or stabilizing feedback to the other.
Ah, so the connections between the modules were the secret sauce.
It seems so. These modular models with the right kind of intermodule connections could replicate the whole pattern. They were robust to silencing just one module, because the intact module could send that corrective signal across.
Makes sense.
But, just like in the experiments, if you simulated silencing both modules at once, there was no healthy partner left to provide the correction, and the model's selective activity failed to recover.
That really strengthens the idea that the actual brain circuitry in ALM isn't just one big homogeneous network, but has this kind of built-in modularity and redundancy. It's like having two engines that can help keep each other stable.
Absolutely.
Yeah.
It strongly suggests that this modular organization, this redundancy, is a core design principle that allows for reliable motor planning, even when biological components are noisy or temporarily disrupted.
Okay, let's try and wrap this up then. Let's synthesize the main insights we've pulled out from these these sources today.
Okay, first I think is that the brain activity in ALM that underpins motor plans is, well, surprisingly tough. It can handle pretty significant brief disruptions as long as they're mostly kept to one side of the brain.
Right. Second, this toughness, this robustness, it isn't just luck. It seems to critically depend on communication from the other unperturbed hemisphere, probably via the corpus callosum, acting as a crucial corrective signal.
Third, the recovery process itself is smart. It's not just turning everything back on. It's selective. It seems to prioritize rapidly fixing the specific activity patterns, that coding direction, that actually matter for guiding behavior.
Fourth, the combined evidence from the experiments and the modeling really points towards the premotor cortex using a modular and redundant design. Like, good engineering, having backup systems interacting seems key for reliable function.
Yeah, exactly. And maybe a fifth point is that understanding this redundancy helps explain something often seen in neuroscience, why sometimes temporarily knocking out a single brain area doesn't completely abolish a behavior. The brain, especially the other hemisphere, might have ways to compensate.
That's a really fantastic deep dive. It paints such a clear picture of the brain's resilience, its internal backup systems, and this dynamic dance between the hemispheres to keep our actions on track.
It really does. And it makes you wonder, doesn't it? Think about these principles: modularity, redundancy, selective recovery. How might these same ideas apply beyond just motor planning? Could they be fundamental to how we, say, hold on to memories over time despite interference? Or how we make consistent decisions when things are distracting us?
Yeah, good question.
And maybe even taking it a step further, what can we learn from these biological solutions? How might understanding the brain's evolved strategies for fault tolerance inspire us to design more robust AI or more reliable robotic systems? It definitely leaves you with a lot to think about, well beyond just how a mouse plans to lick.